All right, thank you so much for being here today. It's my pleasure to introduce the Vanderbilt group that's going to be presenting Grand Rounds today. My name is Emily Glanton. I'm the Associate Director of the Data Management Coordinating Center for the Undiagnosed Diseases Network. And I'm just going to give a brief overview of where things are at with the UDN before we turn it over to the presenters for today. So next slide, please. Just as a reminder, the overall goals of the UDN as set forward by the NINDS at the NIH is to continue the success of the UDN in improving the way that we diagnose participants who are unable to find a diagnosis and doing that with a collaborative method. Another main goal is facilitating research into the causes of these conditions. And then third is promoting a community so that we have a collaborative community that we do this work together with. Just in case you haven't seen the map lately, we have 24 clinical sites across the United States. We also have our research cores and then the Data Management Coordinating Center and the Sequencing Core. There's more information as well on our website about all of the different sites and cores if you're interested in learning more about each of these institutions. And then just to give you an update on the numbers, these numbers were updated in July and our summer quarterly report is out. So if you wanted to dive more into these numbers, that is available on our website as well. We've got just under 9,000 cases that have been submitted and about 3,800 participants who have received a telehealth, in-person, or additional research evaluations at this point. We've identified a diagnosis for 1,054 participants at this point and newly described 97 new disease gene associations. And then just as a reminder for today's Grand Rounds, we would love for you to ask questions throughout, and then we'll ask those of the presenters in the Q&A at the end of the session. And then just as a reminder for this information is presented for educational purposes and can be reused for your own personal non-commercial uses if it remains unchanged, but can't be published in print or electronically without prior written permission from the UDN. And then I'll just share that we will be sharing these Grand Rounds recordings on YouTube moving forward. So those YouTube links will be available for sharing with your network if you want to share and provide the information that was shared today with anyone in your network. And with that, I will turn it over to our presenter for today, Kathy Shear, who is Assistant Professor of Biomedical Informatics, Pediatrics, and Biostatistics at Vanderbilt University Medical Center. We're delighted to have Kathy, so I'll turn it over to you now.
Awesome. Thank you so much, Emily, for that very kind introduction, and good afternoon, everybody. I'm really excited to share with you all some of the recent artificial intelligence innovations for supporting rare disease diagnosis at the Vanderbilt clinical site. Now, before diving into the details, I would like to introduce everybody to someone. So this is Blake. He is an 8-year-old boy from Mount Juliet, Tennessee, and he's got a great matching smile with his furry friend over there. Now, when Blake was born, he was immediately taken to the neonatal ICU, where doctors found complications with his vision, his hearing, and his growth and development. But they couldn't quite figure out what the diagnosis was. Now, this marked the beginning of a very long journey towards finding an answer for Blake. At one point, Blake's mom remembers taking him to go see a specialist on every single floor of the children's hospital. After 8 years without answers, Blake was referred to Vanderbilt and finally received a diagnosis. It turns out that Blake was diagnosed with an ultra-rare genetic disorder that only 25 other people in the world have. The diagnosis was life-changing. Enable connection, share knowledge, and most importantly, proactive care for Blake, who is now thriving in the 3rd grade. Now, I'm sure that for many of us in this audience, that it's certainly no surprise that Blake is not alone. Worldwide, there's an estimated 400 million individuals who are living with a rare undiagnosed disease. The NIH currently recognizes over 10,000 rare diseases, of which 80% have a genetic etiology. Among the 10,000 currently recognized rare diseases, about 550 currently have an FDA-approved treatment. In 2019, the estimated total economic burden of rare diseases in the United States was nearly $1 trillion, with just about half being attributable to direct medical costs. In the United States alone, it's estimated that 1 in 10 Americans are living with a rare undiagnosed condition. And for many patients like Blake, the average diagnostic odyssey can last between 6 and 10 years. Now, the delayed rare disease diagnosis is a $500,000 problem per patient due to avoidable healthcare expenditures, including unnecessary testing procedures, ineffective treatments, and avoidable hospitalizations. Now, all that is to say is that we need earlier and more accurate diagnosis. And given the size of the problem, we need to do this at scale. So Emily showed this slide earlier, and UDN is one of the national consortiums that is tasked with diagnosing and caring for these individuals. And for the next 40 or so minutes, I really want to zoom in on this map and share a little bit about what we're doing at the Vanderbilt clinical site in terms of developing and implementing AI technology to help us with diagnosing and caring for these patients. Now, Vanderbilt has been a champion for rare disease patients and was one of the founding sites of the Undiagnosed Diseases program since 2008. Our institutional diagnostic rate is 62%, and 21% of the diagnoses led to changes in therapy, and 79% led to changes in management. In 2021, we established our own institutional rare disease program that is modeled after the UDN called the Podosnak Center for Undiagnosed and Rare Disorders. And in the last 3 years, we've nearly tripled the number of patients served. And by the year 2030, our goal is to serve at least 1,000 patients annually. Now, scaling up to 1,000 patients per year is certainly no easy feat. Next, I want to talk about some of the barriers to scalable rare disease diagnosis and how those barriers open up opportunities for artificial intelligence. The first barrier I want to highlight is that manual review of patients' electronic health records data, or EHR data, is very time and resource intensive. At our clinical site, it can take up to 3 hours of our clinical experts' time to deep phenotype a patient. I'll explain what deep phenotyping is just a little bit. So the question here is, how can we develop and leverage these AI tools to reduce the manual burden and free up valuable clinician capacity? The second barrier I want to highlight is that the breadth of rare disease knowledge now exceeds any individual clinician's cognitive capacity. So like I mentioned earlier, there are currently over 10,000 known rare diseases, but that's growing at a rapid rate of 300 newly described each year. Therefore, it becomes very challenging or nearly infeasible for any individual clinician to keep up and stay abreast of the field. So the question here is, how do we develop these tools to augment and support clinical decision-making? So let's start by diving into that first barrier. So this slide shows a condensed version of the Vanderbilt UDN site's diagnostic workflow. Now there's a lot to look at on the slide here, but what I want to point everybody's attention to is that phenotyping step on the top right. Phenotyping is the systematic characterization of a patient's key clinical features, and it's incredibly important, not only for informing downstream hypotheses, but also for informing downstream resource allocation. Now, rare disease phenotypes are typically embedded in the patient's EHR data. And the patient's EHR data can be broadly categorized into 2 data types. We have the structured data, which consists of the patient's demographics, billing codes, labs, and medications. But the challenge here is that detailed rare disease phenotypes are often poorly captured by standard diagnostic or billing codes. So we turn to the unstructured information, and specifically the unstructured clinical notes. But as you can imagine, manually combing through pages and pages of the patient's EHR and then extracting these rare disease phenotypes is very time-consuming and difficult to scale. So the question here is, how do we turn this vast amount of clinical text and transform that into something that's more standardized and clinically actionable to support rare disease diagnosis? Now, one of the ways we can address this challenge is to use artificial intelligence, specifically large language models or LLMs. So at our site, we developed Rare-Phenix, which is an end-to-end AI framework for rare disease phenotyping. Rare-Phenix emulates clinicians at 3 key steps in the phenotyping workflow, including phenotype extraction, standardization, and prioritization. So next, I want to dive into a little bit more detail about each of these modules. The first module is phenotype extraction. So the idea is, how do we guide these LLMs, these models, to identify and extract relevant diagnostic phenotypes from clinical notes? The way we can do it, one way we can do it, is by adapting these foundational LLMs to do this downstream task. And we can do this by starting with a set of annotated corpora. And so here we have information from 2 different sources. One are documents from the rare disease database, and also the second source is a collection of synthetic patient notes containing the diagnostic phenotypes that are curated by UDN clinicians. With this annotated corpora, we can then adapt or guide these LLMs using foundational tools such as ChatGPT and Llama to then using techniques such as fine-tuning and few-shot prompting. And what comes out of this process is an LLM that is specifically tailored to rare disease phenotype extraction. So next, really quickly, I want to highlight 2 key paradigms for adapting LLMs to downstream tasks. One very broad approach is fine-tuning. This is a technique where we can adapt these foundational LLMs to rare disease phenotype extraction by training it on additional label data. So the idea is that we would start off with a general purpose LLM like Ernie or Elmo. And with our annotated corpus that consists of the labeled phenotypes, we can then additionally fine-tune it to obtain a rare disease-specific LLM. Now, I want to compare and contrast this with a second paradigm that has been popularized in the last 5 years, and this is called prompt learning or in-context learning. Now, rather than having a label corpus and doing additional training, in-context learning reformulates the specific tasks as textual prompts, so the LLM itself learns to predict the desired output. And so here we can prompt BERT and ask it to extract symptoms. We can ask it to extract signs and output them in a list or extract diseases. Okay. So what does this mean for rare disease phenotyping? So we published a study a few years ago where we had LLMs extract 4 different types of rare disease entities, and we compared fine-tuning versus prompt learning. What we found was that fine-tuning on a label corpus performed better overall in terms of phenotyping accuracy. Interestingly, prompt learning did better on extracting certain entities with just one labeled example. And so what this tells us is that these prompt learning approaches and instruction approaches may reduce the bottleneck of having to rely on manually labeled data. And this is especially attractive for rare diseases where large annotated data sets may be difficult to come by. So back to module 1, the idea is that we would provide the patient's clinical notes. This is not a real clinical note. And then we would pass it through the rare disease phenotype extractor. And then in this case, the output of module 1 is a list of the phenotype mentions. And for this patient, he or she had 3 different phenotypes. Now, moving on to module 2, this is the standardization of phenotypes to human phenotype ontology terms, or HPO terms. And this is what I'd like to call the apples to apples module. Now, human phenotype ontology terms are really critical in genomic medicine for facilitating downstream diagnostic analysis. At our clinical site, HPO terms are used for prioritizing genetic variants. And we also use them for assessing phenotype-genotype correlation using our own institutional biobank, BioView. HPO terms can also be used to find similarly affected patients on platforms such as Matchmaker Exchange. So for module 2, again, we start with the phenotype mentions, and the goal is to map these free text mentions to a structured HPO term. And I won't get into the details of it, but we use a technique called retrieval augmented generation that essentially grounds the LLM's retrieval and generation process in the dictionary HPO terms. Now, switching gears and moving on to Module 3, this is the prioritization of diagnostically informative phenotypes. So this step really hinges on a key clinical intuition, which is that the more specific and the more rare the feature is, or the phenotype is, the more diagnostic insight that it provides. So let me give everybody an example. So if we start with a phenotype such as scoliosis, which is typically a little bit more common and non-specific, it's actually associated with more than 1,600 diseases. But if we go down deeper into ontology, the more clinically specific, the more uncommon and rare the phenotype is in the general population, these phenotypes tend to be associated with far fewer diseases, and that can be really helpful in terms of narrowing down a differential diagnosis. So to emulate this using machine learning artificial intelligence, what we did is that we trained and validated a number of different ranking models using ontology-derived features and also the patient demographics as predictors. So what comes out of this module is a list of re-ranked HPO terms. And in this specific example, the term stroke was deprioritized because it's secondary to another term on this patient's phenotype list. So putting everything all together, Rare-Phenix is a clinician emulating end-to-end AI framework for rare disease phenotyping. So next I want to briefly switch gears and talk a little bit about the performance results for Rare-Phenix. We trained this model on patient data from 11 UDN sites, and we externally validated it on Vanderbilt patient notes. In total, we had just over 2,800 patients. So this slide summarizes the end-to-end performance of Rare-Phenix. Our ground truth consists of the physician-curated HPO terms. And we had 3 different baseline comparators. PhenoBERT, which is a strong deep learning approach. Fast HPO-CR is a very efficient dictionary-based baseline. And we also had a ChatGPT 4.0, which was the state-of-the-art model at the time, where we use a single prompt to really compare it against the modularized architecture of Rare-Phenix. Now, our primary outcome was phenotypic concordance measured by ontology-based semantic similarity. And we also measured a couple of secondary outcomes that are the traditional classification metrics. Now, on the left-hand side, on the x-axis of this graph, we have cutoff K. So this is looking at the top 10 HPO terms, top 20, top 30, et cetera. On the y-axis, we have the metric. Within each panel, the baseline models are depicted using the dashed or dotted lines. And then different configurations of Rare-Phenix are depicted by the solid lines. So what we can see at a glance is that Rare-Phenix consistently outperformed the baseline across all metrics and all cutoffs. We also did a number of ablation analyses to really isolate the performance gain with each additional module of Rare-Phenix. So compared to the baseline alone, having the Rare-Phenix extraction module improved the F1 score, which is a measure of classification accuracy, by about 20% relative improvement. Adding standardization led to an additional increase, and also adding the last prioritization module led to an additional increase, about 20% relative increase in the F1 score. And so what this tells us is that Rare-Phenix tends to rank the relevant HPO terms higher than these baseline approaches and may have the potential to support clinicians' phenotyping workflow. Okay, so I want to pause here for a second. So far, we only assess concordance with clinician-curated phenotypes. But what about potential diagnostic utility? So this question is really getting at, if we start with a list of HPO terms and we use this as input to a downstream disease prioritization tool, for example, Lyrical, what Lyrical outputs is a top K ranked differential diagnosis. And so the idea would be to see whether the true diagnosis, the patient's true diagnosis, would be included in the output. And if it is, it would provide some signal that these HPO terms are potentially diagnostically informative. Okay, so we did this analysis on a subset of our sample of only 29 diagnosed patients. And on the x-axis here, we have the disease rank threshold. So we have the top ranked disease, the top 5 diseases, the top 10 diseases provided by Lyrical. On the y-axis, we have the diagnostic accuracy. So the black bar here represents the clinician-curated HPO terms, and the interpretation of this result is that the true diagnosis was ranked in the top 5 for 7% of patients. And if we look at the top 10 diseases, the true diagnosis was included for 14% of patients and for 21% of patients, respectively. Now, if we look at the other color bars, these all represent performance metrics of the automated methods. Among the automated methods, Rare-Phenix with Llama 270 billion generally achieved the highest diagnostic accuracy, and this was more visible, especially at broader rank thresholds where it outperformed clinician-curated HPO terms. But again, we interpret this with a lot of caution given this modest sample size. Another interesting finding is that Rare-Phenix also captured disease-related phenotypes, about 5%, that were missed during manual curation. And so what this tells us is that these AI tools may have the potential to improve phenotyping completeness and supplement the clinician's workflow. And currently, we're working on a prospective evaluation at the Vanderbilt site, and this work is led by a PhD student, Mike Enger. Okay. So that was a little bit about AI for phenotyping. Next, I want to switch gears and talk about that second barrier and how we can leverage AI to support clinical decision-making. Now, rare disease diagnosis, especially at the UDN, often involves multidisciplinary evaluation. As I mentioned earlier in the talk, the rapidly expanding landscape of rare diseases makes it very challenging or nearly infeasible for any individual clinician to keep up and stay abreast of the field. So with that in mind, we asked the question, what if we gave artificial intelligence a seat at that conference room table? So we investigated this question through a number of different studies. So this was a study where we compared a widely used differential diagnosis tool, Exomizer, on 424 UDN patients. As input, we provided the patient's HPO terms, and ChatGPT 4.0, which was a state-of-the-art model at the time, outperformed Exomizer in terms of differential diagnostic accuracy. We had another study that was published last year where we compared LLM's diagnostic accuracy against historical UDN clinical review rates. And what we found was that ChatGPT 4.0 achieved twice the diagnostic accuracy than historical UDN clinical review. And the LLM also provided a helpful differential in 23% of cases. And helpful here is defined as either the LLM getting the diagnosis exactly right or getting a very closely related diagnosis. Differential generation was both cost and time efficient. So it cost us about 3 cents and 5 seconds of processing time per patient. So these results suggest that AI has the potential to serve as a partner for rare disease diagnosis, that we can leverage these early in the diagnostic workflow to suggest plausible conditions that we may be able to support physicians to help them diagnose patients more accurately, more efficiently, and more cost-effectively. So currently, we are using these LLM-generated differentials right prior to the patient's clinic visit, where the physicians will review them. And this pipeline has been implemented, deployed at the Vanderbilt clinical site for the last 2 years. And I'm really excited that it has been able to really serve more than 150 rare disease patients. We're also formulating and planning a prospective study. Okay, So for the next part of my talk, I want to shift gears and talk a little bit about some of the more recent work that we've been doing at the Vanderbilt site, specifically on building frontier AI agents for rare disease diagnosis. And these ideas really hinge on the paradigm shift where we move away from pattern matching in these AI models towards really unlocking and maximizing clinical reasoning in these modern LLMs. So before I dive into the details, I think we can do a little bit of a quick exercise just to switch things up. So for all the clinicians or clinicians in training in the audience, go ahead and read that question on the slide and think to yourself what the correct answer is. So which medication reduces mortality in systolic heart failure? I'll give everybody about 3 seconds. Okay. So once you have an answer, go ahead and look at that same exact question on the right, and then look at the answer choices and think to yourself what the correct answer is. I'll give everyone 3 seconds. Okay. So I'm sure most, if not everybody, picked C as the correct answer for both questions. And really the difference between the question on the left and the right is that the correct answer, ACE inhibitors, was swapped out by none of the other answers. Now, just about a little under a year ago, Betty et al. Actually did a very interesting study where they sought to understand whether LLMs can reason like clinicians. So they did the exact same exercise that we just did, where we have the original multiple choice questions, and the authors then ran a separate set of experiments where the correct multiple choice answer was replaced by none of the other answers. The hypothesis is that if these models could truly perform clinical reasoning rather than just pattern matching, then their answers should remain the same. And they tested this across a number of state-of-the-art LLMs at that time. And what they found was that all of the models actually performed worse on the none of the other answer questions, revealing that they tend to rely on pattern matching rather than true clinical reasoning. So if we fast forward about 10 months, so this paper was recently published in Science, where the researchers essentially evaluated frontier LLM's performance on reasoning tasks of physicians. And their conclusion was that in all experiments, the LLM outperformed physician baselines and displayed continued improvement from prior generations of these models. And so the field is definitely moving at such an accelerated pace that these LLMs are definitely displaying and demonstrating capabilities that can really match physicians' reasoning. So reasoning, by no doubt, is definitely very critical. But when it comes to rare disease diagnosis, we also require a process. So the challenge is not just the reasoning model itself, but understanding what evidence to gather, how to interpret phenotypic data, genetic data, how to weigh the information, and when to revisit assumptions and pivot if findings are unrevealing. So the question is, can we give these AI models a diagnostic process that clinicians can inspect, critique, and improve? Now, the solution that we've come up with to answer this question is to build a diagnostic policy layer around the reasoning LLM. So what this diagnostic policy does is that it guides the model through an end-to-end diagnostic workflow. And we also provide a suite of biomedical tools that are specific to rare disease diagnosis, such as information to assess phenotype gene association, gene disease association, and also information related to genetic variant data as well. We also provided access to the literature so that it can search most up-to-date information to retrieve the relevant evidence. So one important thing to note here is that because the diagnostic policy is external to the LLM, it can be ported, audited, and revised. So imagine if we have the same policy, and let's say we have 2 different models, the same policy can be ported across different LLMs. Now, suppose we're at the UDN, we have 24 sites. Each site may have their own local diagnostic workflow. And so what we can do is take that same policy and we can revise it and adapt it to another site. So the policy, the key point is that it can be updated as clinical knowledge and standards of practice evolve. So the way that we operationalize the policy-guided reasoning is through LiteOdyssey, which is a lightweight AI agent that we developed at Vanderbilt site for policy-guided rare disease diagnosis. And this work is led by a PhD student at Vanderbilt, Han Nguyen. So the idea here is that LiteOdyssey requires only a single reasoning capable model. So really any modern frontier LLM that we use, like ChatGPT, Gemini, Claude, etc., or DeepSeek. The model uses the human expert guided policy, which I mentioned earlier, and that policy is portable across different models. LiteOdyssey is lightweight in that it doesn't require additional fine-tuning or very intensive local architecture, such as multi-agent system coordination or external databases. What it takes as input is the list of patients' HPO terms or a narrative text, such as a snippet from their clinical notes. LiteOdyssey can also incorporate genetic variant information if available. So next I want to highlight three key components of LiteOdyssey. First is how that policy is built. So this is built through an approach that we coined policy iteration with human feedback, or PIHF, in which clinicians would review the AI model's reasoning traces. It would correct recurrent diagnostic errors and then optimize the model's performance. Now, in traditional machine learning, when we're speaking about optimization, typically, one way we can think about it is that we're optimizing or updating a model's weights. But here in PIHF, the object of optimization is a diagnostic policy. So we would optimize that policy, wait for the performance to stabilize, and then freeze that policy for downstream evaluation. Now, the second key component of LiteOdyssey I want to highlight is the tool-grounded adaptive reasoning loop. This is, again, the policy equips the model with a suite of biomedical tools. And what it does is it performs an adaptive reasoning loop where it reasons about the patient's case. It can call different tools, read and digest the evidence, and then decide the next step, and then repeat the process. The third thing I want to highlight is that the output of LiteOdyssey includes a rank differential diagnosis with clinician audible reasoning traces, so that every suggestion is supported by very transparent evidence and also an audit trail. So just to give everybody a more intuitive understanding of what this tool does, I'm going to pull down the slides and show everybody a quick live demonstration of the tool. Okay, so here is essentially the user interface of the LiteOdyssey tool. Here I'm just going to provide a case. This is not a real patient case. This is a young woman who has had years of unexplained nausea, early satiety, abdominal bloating, pain, severe weight loss. She has had repeated hospitalizations. The routine GI infectious and autoimmune evaluations have been unrevealing. So we're gonna send in this prompt, and so the LiteOdyssey tool will kind of get to work. And what we can see, I really just want to highlight two key points in this demo is that the first key point I want to highlight is that every single step of LiteOdyssey's reasoning process is human readable and auditable. So you can really, follow along and see how it's making those decisions, including what types of tools it's calling, and how it's arriving at its differential diagnosis. And for some of the more technical folks on the call, you may see that throughout this process, the model is also running bash commands where it's calling these tools that I mentioned earlier. So here, this is an example where it's calling the Feed-to-Gene Monarch Initiative tool, where it's searching up different HPO terms and validating them. Okay, so in the interest of time, I'm just going to show everybody the final result that was pre-run and generated. So what LiteOdyssey will generate is a structured clinical diagnostic report for the user's consideration. So what it provides is first the key phenotypes converted into HPO terms, and also provides a differential diagnosis. In this case, this synthetic patient, is suspected to have a very rare mitochondrial DNA depletion syndrome. So here, as you can see, the output is, again, very source traceable. So the model provides essentially all of the reasoning, including additional external information and also evidence that I retrieved from other sources, such as the biomedical literature. And then section 4, again, lists the different types of evidence, and it tells the user exactly where that evidence was sourced from. It provides some additional information about a couple potential other diagnoses. And it also provides a calibrated confidence score, and it also tells the user what some of the limitations are. And at the very bottom, it provides a list of recommended next steps for the user's consideration. Okay, so now I'm going to flip back to the slides. And next, I want to dive into a little bit more detail about the performance of this tool. So LiteOdyssey outperformed frontier LLMs in diagnostic accuracy across 2 different public data sets, Lyrical and Phenopacket. So here, LiteOdyssey is in blue. We can see that it is outperforming 4 different state-of-the-art baseline LLMs without any type of policy guidance. One really interesting finding I want to highlight for this audience is that with that policy guidance, a smaller open weight model, Quen 35 billion, actually outperformed, in some cases, outperformed or even matched the performance with very much larger scale frontier closed source models like DeepSeek Flash, ChatGPT 5.4, or DeepSeek 4 Pro. And so this really demonstrates the portability of that diagnostic policy. Next, we also performed an evaluation on 515 UDN cases. We had physicians independently adjudicate the model outputs using a validated scoring scale. This is a scale from 0 to 5, where 0 is the differential is not helpful at all, to unlikely to be helpful, all the way up to 5, which is where the differential includes the exact diagnosis. So a couple of key points I want to highlight here is that first, LiteOdyssey was more likely to include the exact diagnosis in its differential than ChatGPT 5.4 without any policy guidance. Now, on the other end of the spectrum, LiteOdyssey was also less likely to generate unhelpful differential diagnoses compared to GPT 5.3, and these results are all statistically significant. So really quick takeaway is that rare disease diagnostic reasoning we've demonstrated through LiteOdyssey that it can be externalized into an AI reasoning policy that is human readable, auditable, and portable across different models and data sets. And we're also planning to conduct a prospective study at our site to rigorously measure the impact of this tool on clinical and patient outcomes. Okay. So for that first half of the presentation, I think we've seen a lot of exciting applications of artificial intelligence. But for the next 10 minutes or so, I want to switch gears and talk about how we can think about rigorously evaluating these tools' reliability for clinical deployment. So next, I want to highlight a study that we recently did on developing a statistical framework for evaluating the repeatability and reproducibility of large language models. So when we look across the literature, there's definitely growing evidence demonstrating LLM's accuracy for diagnosis. But accuracy alone doesn't reflect how reliable the model's outputs are. So I think it's important before we go any further to understand how these models are generating output. The way that they're generating tokens or the next word is actually by sampling from a stochastic probability distribution given prior context. So it's possible that when we have the same prompt, same parameter setting, same model, same everything, that when we run the model twice, that's possible that would give us a different output. So here we have a prompt about a patient. We ask it to provide the top 3 possible causes. And in the first run, the model actually got the right diagnosis, meningitis, but it didn't even include it in the second run. So in high stakes applications like diagnostic support, the consistency of the LLM's output can have an impact on downstream decision making. There's also growing guidance from journals and also the FDA on the importance of measuring LLM variability. The FDA specifically urges researchers and developers to quantify the uncertainty by assessing the repeatability and reproducibility of AI outputs. Now, the FDA defines repeatability as the closeness of agreement of repeated measurements taken under the same conditions. So we're talking about the same LLM, same parameter setting, same prompt, same everything. Reproducibility is similar, but now we're looking at repeated measurements taken under different pre-specified conditions. So that could be different prompts, different users, or different UDN sites. Now, one thing I think is really important to think about is that unstructured data really challenges the traditional notions of repeatability and reproducibility. So if I ask everybody on this call to, look at these 2 outputs and ask yourself, are the following 2 outputs the same? What does everybody think? It's pretty hard to say, right? So I think on the surface, when we look at the wording and the phrasing, the outputs look pretty different across the 2 runs. But if we look more closely at really the meaning of what it's trying to get at, it's really telling the clinician that, hey, meningitis is the likely diagnosis. So when the outputs differ in wording, but not in meaning or semantics, how do we define these measures, the repeatability and reproducibility? Beyond semantics, I also want to highlight why the LLM's internal stability matters. So here we, again, now have 2 outputs and they look very, very similar on the surface, but it's also possible that the underlying probability distributions can differ substantially across runs. So in the first run, the LLM was actually pretty likely to output meningitis. But when we look at the second run, it's actually very uncertain and equally likely, just about equally likely, to also provide some of these other possible diagnoses. So this poor internal stability is a reflection of the model's unreliable reasoning. And in high-stakes tasks like diagnosis, how the model reasons matters just as much as the answer itself. So in this project, we had a twofold objective. First is to develop a framework to quantify the repeatability of LLMs. And within repeatability, we also quantify both the semantic and internal repeatability. Similarly, we also want to quantify the reproducibility across different experimental conditions. In this case, we're using different prompts. And within reproducibility, we also develop ways to quantify semantic and internal reproducibility. Okay, so I won't dive into the details of the notation and setup, but really the takeaway is that we developed a set of metrics, 4 to be exact, that researchers can use to systematically quantify and evaluate the repeatability and reproducibility of large language model outputs. So next, I want to briefly talk about the evaluation strategy. So to assess the repeatability and reproducibility of LLMs, we evaluated 4 different models across 5 different reasoning prompts on 2 data sets. And for each prompt case LLM combination, we ran 100 independent runs. Now, this slide provides an overview of the 5 different diagnostic reasoning prompts that we use, and these range from traditional chain of thought prompting to differential diagnosis prompting to Bayesian reasoning chain of thought prompting, which involves dynamically updating the diagnostic likelihoods using new evidence. We use two evaluation data sets. The first data set consists of the USMLE standardized exam style questions. And the second data set is a real world data set from the UDN of 90 patient cases. So on this slide, I really want to quickly summarize the results. On the x-axis, we have the semantic repeatability score. On the y-axis, we have the internal repeatability score. Each vertical column represents a different model, and then each row represents a different data set. Within each panel, we have these different color lines and contours that correspond to a different diagnostic reasoning prompt. So one takeaway here is that in general, semantic and internal repeatability varied across different prompts, models, and data sets. For ChatGPT-4, the Bayesian chain of thought prompting had higher semantic repeatability across both data sets, which suggests that it could be more semantically stable in this setting. Next, we also investigated whether diagnostic accuracy was related to variability in the LLM's outputs, and we assessed the relationship between repeatability and reproducibility scores on USMLE cases stratified by the LLM's diagnostic accuracy, so correct versus incorrect. So here on the x-axis, we have the semantic versus the internal repeatability score on the y-axis. And really the takeaway here is that LLM repeatability is generally not correlated with diagnostic accuracy because we see a lot of overlap between the blue and the orange contour plots. And the same takeaway also holds for LLM reproducibility. So really the key thing to note here is that accurate diagnoses do not necessarily correlate with consistent outputs across different prompts. So what that means is that an LLM getting the diagnosis right once doesn't mean that the LLM is going to consistently get it right. So to quickly summarize, in this work, we provide a generalizable FDA-aligned framework for evaluating LLM output variability that's agnostic and can be applied across different models, prompts, datasets, and tasks. Repeatability and reproducibility were generally not associated with diagnostic accuracy, meaning that an LLM could be correct once, but unstable across repeated runs, or it could be consistently wrong. This work can support more rigorous study design when LLM outputs are used as study endpoints. And as a biostatistician by training myself, I think this could really pave the way for more formal statistical power calculations in clinical studies as we move towards evaluating these tools in the clinic. So in the last 2 minutes, I really wanted to conclude with a couple of lessons learned from my group at the Vanderbilt site when it comes to building AI tools that fit UDN's clinical workflow. The first lesson is the importance of starting with the clinical workflow and not the model. So understanding what decision or task does the AI tool support, where does it fit in the clinical workflow, and who is the intended user are all really critical to figure out before we think about what model to develop or what methods to use. The second lesson I want to highlight is really the importance of evaluating early, repeatedly, and prospectively. And I think these tools are advancing at such an accelerated rate that really outpaces our ability to evaluate them in clinical settings. And I think the UDN can serve as a very valuable testbed for rare disease AI that can really move the field from retrospective validation to prospective human-centered evaluation so that we can understand how to better leverage these tools to provide better care for all patients. And so with that, I would like to thank the patients for participating in the UDN, our funders, and also the entire Vanderbilt UDN team. And last but not least, thank you all so much for listening. I'd be happy to take any questions. Next, I think I'll pass it over to Emily.
Thank you so much for a great presentation, setting the stage for the problems that we need to solve and some of this work. So we've got a few questions that have come in through the Q&A. And just a reminder, any questions that you have, please do put those in the Q&A so we can answer those. So the first question for you, Kathy, is from May Malikton at the NIH. Great presentation. I'm curious whether you've looked beyond overall diagnostic performance and stratified results by discovery type. So, for example, how do the Rare-Phenix or LiteOdyssey perform in cases involving novel gene disease discovery, phenotypic expansion of known disorders, new disease mechanism, gain of function versus loss of function, dominant negative effects, or novel inheritance patterns?
Thank you for that question. That's a really great point. For those two studies, we haven't done a stratify analysis, but I think now that you've suggested, I think there's a lot of value in that. So that's definitely something that we're going to look into. We're also, for LiteOdyssey, I think the idea of that diagnostic policy being portable to different diseases, I think opens up different opportunities for being able to adapt it to different types of diseases. And so that's something on the horizon as well. So thank you for that question.
Thank you. And then the next question from Jonathan Sheehan, fantastic talk. When testing accuracy, how do you ensure that none of the training data was included in the testing sets?
Yeah, that's a great question. So what we do is that we do essentially a patient level split when we're doing the training validation. So all the trainings patient data is essentially locked, and then we only evaluate on a separate holdout data set. So there's really no contamination or crosstalk between the training and validation data. Yeah.
Great, thank you. And then from Pankari Singhal at University of Pennsylvania, a question about if you can just share more about what the clinical policy the model sees looks like.
Yeah, that's a great question. So the clinical policy that the model sees, one, it's you can think about, I would say there are two different components to it. The first component is essentially a document that's written in natural language that essentially consists of a list of instructions that steers the model and kind of guides it through a structured list of reasoning steps. And we develop those steps with collaboration from our clinical experts to make sure that that's really grounded in rare genetic disease diagnosis and really kind of mimicking their workflow. Even though we have that document, it is not deterministic. We still give these reasoning models some leeway where they can decide how to dynamically kind of navigate the process depending on how much information, how much evidence is accumulated at a particular step in the reasoning process. The second part of the diagnostic policy, in addition to that natural language document, is the set of biomedical tools that we engineered. And we purposely made sure that these are open source tools that folks can use. So we essentially built them in and then incorporated that with the natural language document so that the LLM can essentially decide which tool to call at which step of the reasoning process. So thank you for that question.
Wonderful, thank you. And then another question is just if you can share a little bit about throughout all this many years of work now that you've been working on these problems, what has been, in your opinion, the hardest problem to solve?
Yeah, I think the hardest problem to solve is really, I think what makes the UDN so special is that these are, patients that have been on these very, very long and isolating diagnostic odysseys and, can probably see the UDN as one of their last resorts. And so I think that the patient population itself is a more diagnostically challenging test bed. And I think really the challenge and I think really the opportunity is really trying to think about how we can maximize the capabilities of these AI tools and to train them and to adapt them in a way that they can be successful on to really solve some of the most medically challenging cases that we see in medicine. Yeah.
Great. Yeah, that ties in a little bit to May's question about not only having it accurately pick out a diagnosis that does exist, but helping us to discover new conditions that we're discovering.
Yeah, that's a great point.
All right. Well, we've answered all of the questions that we had. So thank you again for this great presentation today and for all of this work. And just for anybody who's interested, our next Grand Rounds will be in October, so you can register at the link on our website there. And with that, I will close this out for today. So thank you all so much, and we'll see you next time.
Thank you.